Hi everyone, my name is Kristianna and I'm the co-president of Reach Out Teach Out at UCI. Welcome to the Reach Out Teach Out Science for Young Scientists series. Today, I'm going to be discussing the importance of designing proper controls for scientific experiments. This presentation is part of our experimental design series, so if you have additional questions about designing experiments, be sure to stay tuned for future videos. In any scientific experiment, there are two basic types of variables. The independent variable, which is what you as the experimenter are changing, and the dependent variable, which is the change that you are measuring. Let's say, for example, we are performing a simple experiment on the role of a chemical compound, which we can call in our case chemical A, on plant growth in a plant such as this one shown here. We decided to hypothesize that chemical A enhances plant growth. But how can we be certain that any changes in plant growth, which would be our dependent variable in this case, are coming directly from chemical A, which is the independent variable, and that they're not due to some unknown factor, which in science we call the confounding variable? Essentially, our question is, how do you determine if an effect is real and not from some unknown factor? The answer is that you have to use controls. The definition of a scientific control is a comparison, and it's used to minimize the effects of confounding variables, those unknown factors that we talked about, to ensure that changes seen are a direct result of the independent variable or the thing that you're adding. Appropriate controls increase the likelihood that the effect that you observed is directly related to the independent variable and not another confounding variable. If you include good controls in your experiment, you can more likely trust your results, which is exactly what we want to see in science. There are two general types of controls, the positive control and the negative control. We will go into a lot of detail on these, but generally, the negative control is the control that has no effect on the dependent variable, whereas the positive control has a known effect. In the next few slides, we'll go into more specifics about these two. The first type of control is a negative control. A negative control is a control that should have no effect on the dependent variable. In our plant growth experiment, for example, we could compare the experimental group, or the group over here that was treated with chemical A, which is dissolved in water, to the negative control group that is untreated and therefore given water without any chemical A. If our hypothesis is that chemical A enhances plant growth, we should see an increase in plant height in the experimental group with chemical A in comparison to the negative control group. If, however, we see similar growth, it will suggest that chemical A does not in fact have an effect on growth. We can then perform a time course and plot the height of the different plants of the water and chemical A groups to see the effect of chemical A on plant growth. If we graph the plant growth over time, it does in fact appear that chemical A enhances plant growth in comparison to water, our negative control group, because we see that at all time points except for day 0, it has a higher height than the water-only group. It is equally important to include positive controls in any experiment. As you may remember, a positive control is a control that it has a known effect on the dependent variable. Positive controls have a couple of different purposes. The first is that it ensures the experimental conditions are optimal. This also helps determine whether the independent variable has a real effect on the experiment, as well as the magnitude of that effect. In the example of the plant growth experiment that we've been talking about, a positive control could be the addition of chemical B instead of chemical A. And this is important because chemical B has previously been shown to enhance plant growth in comparison to a no treatment or water group. If the addition of chemical B to the water does not enhance plant growth compared to the negative control, we can conclude that the conditions used for the experiment is not optimal. For instance, we might have forgotten crucial nutrients for plant growth, or might not have given the plants enough water, which then allows us to troubleshoot our experiments and know what to do better in the future. In this case, we see that the lines for water only and chemical B are pretty much overlapping, which probably tells us that our experimental design is not optimal. We can also use positive controls to compare our experimental growth to the positive control to see the effect size of the chemical, to see if it is less than, equal to, or greater than that of our positive control. In this case, we see that chemical A does in fact enhance plant growth, but not to the same magnitude as chemical B. It is a lower effect. Positive and negative controls are usually not an either-or situation. In most experiments, you should actually have both, just like in our plant growth experiment, where we had the negative control, which was the water only, and the positive control, which is chemical B. It is also important to note that you can actually have more than one positive control. For example, in the plant growth experiment, there may be several different chemicals that are all known enhancers of plant growth to different degrees. If you want to know exactly how chemical A compares to each of these chemicals, it is best to use each as its own control group. So in this case, in addition to chemical A, which was the one that we were hypothesizing enhances growth, we also included chemical B, chemical C, and chemical D. We can see that chemical A seems to outperform chemical C because chemical C is lower, but not as strong of an enhancer as plant growth as chemical B or chemical D. So in conclusion, controls are essential for any scientific experiment because they increase the likelihood that the effect that you are observing is directly correlated to the independent variable that you are changing, and not due to other confounding effects. Negative controls act as the no treatment control, and they show what should happen if there's no effect. In contrast, positive controls are controls with known effect. They're usually used to ensure the experimental conditions are optimal, and they're also used to evaluate the magnitude of the effect.